Alright, so we're going to begin our discussion on cellular respiration and metabolism. So essentially we're going to be looking at how we take nutrients and turn them into energy. And for the cell, energy is going to be in the form of ATP, adenosine triphosphate. Okay, this means there are 3 phosphate groups bound in a row. Okay, and this is basically the gasoline or the fuel that the cells will use. Okay, they don't directly use glucose as energy. They will take glucose or amino acids or lipids, fats, and use that as energy to make ATP. So to look at this, we got to look at what's called bioenergetics. We have to look at the energy in these molecules, and these biological molecules. So when we look at certain reactions, and certain reactions are what we call endergonic reactions. And you can see there is free energy. There's energy in these molecules, because remember, these molecules have bonds, and these bonds have energy. Okay? And so there is usable energy in the bonds in each molecule. In some of these reactions, they're called endergonics. Okay, the first law of thermodynamics states that basically energy cannot be created, destroyed. So energy has to come from somewhere. And if we look at these reactants, okay, here is that potential energy going up here of the molecules. Okay, this is called kind of the usable or the free energy a molecule has. Okay, it is the potential energy, and it's the energy in those bonds. And you can see that potential energy for the reactants is down here. The potential energy or the free energy of the products is greater than the reactants. Okay, this can't just happen. This reaction goes without an addition of energy. So we have to put some form of energy in to make this reaction take place. Okay, to go from a reactants with lower free energy to a product with higher free energy. And this is what happens in an endergonic reaction. Is energy is put in the reaction, and the product ends up with higher usable energy or potential energy. Okay, so in endergonic reactions, okay, again, the products contain more free energy than the reactants, okay, and it requires an input of energy. Okay, energy, this doesn't just appear, we have to put it in to get to a higher usable free energy in the products. Okay, so endergonic reaction, and this is going to be when we're making ATP. ATP is making of ATP is going to be an endergonic reaction. Okay, we take a molecule of ADP, and we'll look at the structure of this in just a second, or just in a couple slides here. But we take ADP and phosphate, inorganic phosphate, and we make ATP. Okay, so here's a little phosphate, bound to a phosphate, bound to a phosphate. Okay, and here's adenosine. Alright, so we have ATP, we have 3 phosphates bound to each other. Okay, so we basically created this bond here, and this is a very high energy bond. We started with just 2. Okay, so we added this 3rd phosphate bond when we create ATP. Okay, and to do this, we had to put energy in to make this take place. So this is an endergonic reaction. Okay. And now looking at exergonic reactions, kind of reverse of what we just saw with inorganic, is the reactants have a relatively high potential energy or free energy. Okay, there's a substantial amount of energy in these bonds. We get our activation energy and our reaction runs, and our products are at a lower free energy. Okay, and so to get from here to here, energy was released. Okay, and this is essentially what we're going to be doing with when we break down glucose. Glucose is going to be broken down into smaller molecules, and ultimately we're going to have release of energy. So you can see we start with glucose, we're going to break down to water and carbon dioxide, and during this we're going to release energy. Okay, and so we can take exergonic, we can take the energy in these exergonic reactions and take it to basically fuel what we just saw, that endergonic reactions. Right. So in this case, we're going to be taking to break exergonic reactions, the breakdown of glucose or lipids or proteins, and we're going to take the energy released of breaking down these molecules, and we're going to take that energy and bring it over to the endergonic reaction of making ATP. And here's kind of just an overview of the breakdown of glucose we'll be doing. Remember, breakdown of glucose is endergonic. We start with a molecule, sugar, glucose, with a higher free energy or higher usable energy, and we're going to break it down to these products, ultimately CO2 and water, molecules that have a lower free energy. But when we break down glucose, we're not going to be doing it in this one large step. We're not going to break down glucose and basically, looking over here, I'm taking glucose and breaking it down in one step and releasing all this energy in one burst. We're going to be taking these little steps, okay, releasing a little bit of energy, a little bit of energy, a little bit of energy, and a little bit of energy. And basically we're going to store this energy so we can use it to make that endergonic reaction required to make ATP. Okay, so again, we do it in these little steps here. Boom, a little bit of energy released each time we're breaking. So we have all these intermediate molecules. It's not just glucose straight to carbon dioxide and water. We have all these little intermediate molecules in the step, and each way we're releasing a little bit of energy that we're going to store and use to, again, make that ATP. So here's kind of the flow of that energy. You can see here, we're going to take food, glucose, lipids, those fats, the proteins, and we're going to take them from higher energy to lower energy, okay, and we're going to release energy, and that's actually going to fuel that endergonic reaction that is going in the opposite direction, making ADP to ATP. Okay, so this cog moves this cog up to higher energy at ATP. Okay, and here's looking at that molecule. Okay, there's ADP, adenosine diphosphate. Okay, 2 phosphates. These little yellows are the phosphate group. So all we're actually doing is adding this phosphate to create ATP. Okay, we're just creating this bond right here. That's the endergonic reaction. We're going to break down glucose in those little steps, store the energy, and then we're going to use that energy to fuel this endergonic reaction right here. Okay, and this is, you'll see, because this is a very high energy bond. So this is a good molecule for the cell to use energy, because when they break this bond, it is a high energy bond, and the cell can use that energy to fuel the stuff that needs to go on in the cell. Okay. So to go forward and start looking at those breakdown steps, those steps to break down, our glucose, our lipids, and so forth, we have to look at a type of reaction, because we will be seeing this throughout those reactions, those breakdown reactions, and also the reactions to make ATP. We're going to see there are these reactions called oxidation reduction reactions. Okay, again, these are coupled reactions. Okay, if you oxidize something, you're reducing something else. Okay, so they always coupled together. But what is a reduction reaction? Reduction means that one molecule is gaining electrons. Okay, in this case, we're talking a pair of electrons, 2 electrons. Okay. I like to think when you gain negative, you're adding a negative, you're reducing the number. So think of, if you want to think about it that way, reduction means that an atom or molecule is gaining electrons. And we will see that it doesn't gain it by itself. Usually those electrons don't come freely, they come combined with hydrogen atoms, hydrogen ions. Okay, so whenever you see the molecule, we'll be talking specifically one, catamine, adenine, dinucleotide, NAD. When you see a gain hydrogen ion, it also has gained a pair of electrons. Okay, so going from NAD to NADH means it's been reduced. Okay, the opposite, whenever you reduce something, those electrons came from another molecule, and that molecule was oxidized, went through oxidation, because an atom or a molecule loses an electron. Okay, so you can see how these reactions are coupled, because if one molecule is being reduced, something else has been oxidized. Okay, the electrons don't just come out of thin air, they're coming from another molecule. Okay, it's called oxidation, okay, because of oxygen, but it doesn't always mean that oxygen is involved. But oxygen is going to be one of the main players in these oxidation reduction reactions when we start seeing the breakdown of glucose or these nutrients. But they are always coupled. When something gets reduced, something else has been oxidized. The one molecule has passed on its electrons to another molecule. Okay. And we can this sounds in reverse, but an electron donor, a molecule that donates, is a reducing agent, because it's going to reduce whatever. But this reducing agent actually becomes oxidized, right? It becomes oxidized because it's reducing another molecule. And an oxidizing agent is one that receives electrons. Okay, so this oxidizing agent actually gets reduced because it is taking on the electrons, but it is oxidizing whatever molecule is giving off the electrons. Okay, so these reactions are always coupled. We're going to pick up electrons when we break down glucose. We're going to pick up electrons. Right? How do we pick it up? Many times it's going to be picking these electrons up from NAD. NAD is going to get reduced to NADH. And then that means one of those molecules in the reactions was oxidized, lost a pair of electrons or gave off a pair of electrons. Okay, so always coupled. Oxidation reduction reactions are always going to be coupled. Okay. Through these reactions, we're going to see many of the molecules can be both oxidizers and reducers in a chain reactions. Okay, so basically these electrons are going to be picked up and then pass along, picked up, pass along. Okay, and again, one of those main players is going to be that NAD. Right, this is a coenzyme. If you remember from our enzyme lecture, NAD is a coenzyme. It'll pick up an NAD, but then in another reaction, it may get reduced back or oxidized back to NAD. But this doesn't happen by itself because the electrons that are given off, it will reduce some other molecule. Okay, so we're going to see this along the way in the reactions. One of the great electron acceptors to me is going to be oxygen. We'll see why oxygen is important. That's why we breathe oxygen basically, because it is a great electron acceptor. And at the end of this whole day, it's going to accept electrons, okay, to keep the process of breaking down glucose going. Okay, and like I said, usually free electrons are not passed along by themselves, but hydrogen atoms are carrying the electrons. So typically, as we saw with NAD, when it picks up electrons, it also picks up a hydrogen atom. Okay, so this, you can see, there is also electrons accepted here by NAD. Okay, so molecule gains electrons and also gains hydrogen. Molecule loses hydrogen, it is oxidized. Okay, so you can see NAD loses its hydrogen, a little positive there, and it has also lost its electrons or given off its electrons, going from reduced to oxidized. So here's some of our main coenzymes. Remember, these are small, they're larger biological molecules, so they're not cofactors. Remember, our cofactors are just our metal ions, but these are larger, still small molecules that are required for many of these reactions to take place. So we got NAD, nicotinamide, adenine dinucleotide, and flavin adenine dinucleotide. But you can see here is NAD, it's oxidized state, so it hasn't picked up electrons, and NADH. Remember, the electrons usually come carried by hydrogen ions. Okay, you can see it's picked up a pair of electrons. FAD also accepts 2 pairs of electrons. When it accepts its pairs, it picks up 2 hydrogens. Okay, this doesn't mean it picks up 4 electrons. Again, it picks up 2 electrons. So here is 2 electrons, and here we've picked up 2 electrons. Okay, and so I like to think of these reactions. We're going to see this taking place throughout many of these reactions. When we break down, as we break down glucose, NAD is going to be picking up electrons. Think of it as an electron carrier. I like to think of these reactions as harvesting electrons. When we're breaking down glucose, we're harvesting electrons for energy to use in further reactions. Use it as energy later. Okay, so these are kind of like the fruit pickers of the bunch. The NAD and the FAD are the fruit pickers. They're harvesting, instead of picking fruit, they're harvesting or picking the electrons up. Okay, because they're going to pick them up and then they can drop them off later, and we can use those electrons for energy. So here's looking at how the coupling of reduction and oxidation reaction takes place. We have a molecule X, and X is going to donate or lose its electrons. Okay, remember those electrons usually come with hydrogen atoms. Okay, so here X is being oxidized. It has lost its electrons. Who's picked up the electrons is the NAD. Okay, we went from NAD to NADH. So this has been, NAD has been reduced. Okay, so one molecule gets oxidized, another gets reduced in the reaction. NADH can later go, and now NADH can be oxidized. So it's lost or donated its electrons, and now another molecule has picked them up. So Y here has now been reduced because Y picked up the electrons that were released by the reduced NADH. Okay, so these are always coupled. These oxidation reduction, the electrons don't just come out of the blue. They're given from one molecule gets oxidized, and then another molecule in turn gets reduced. Okay, so they just get passed along. These electrons get passed along from molecule to molecule. Okay, but the way we're going to pick up most of the electrons along the way when we're breaking down is picking them up in this NAD molecule. So in a nutshell, this is what we're going to be looking to. I always make the joke of, all right, you got to memorize all of this, but please don't memorize all of this. We are going to be going through a lot of this. This is going to be our glycolysis here. This is going to be our citric acid cycle. This is going to be our breakdown of fatty acids. Okay, so we're going to go through this, but we're not going to, you don't have to memorize all these structures. I know it looks burdensome and cumbersome, but we're going to look big picture on what's taking place through these reactions to look at how we are making this ATP and how it relates to metabolism. Okay, so we're not, you can see how complicated this all can get, but we're going to keep it rather big picture looking at things. But here you can already see these reactions with our NAD taking place. Okay, but these are the reactions involved in breaking down the different nutrients to ultimately harvest the energy to make ATP. Okay, if you just looking forward, if you ever have to take biochem or so forth, plan on memorizing a lot of this stuff. Okay, but for our sake, we're going to be going big picture, looking at kind of the, not the real fine details, but looking at details in where we're getting our energy from in these reactions. So our metabolism in our body, we got two types of reactions taking place. We have anabolism, requires input of energy to synthesize larger molecules. So this would be this, and you can see we're having to put energy in to make these smaller molecules into larger molecules. Okay, a form of anabolism would be making ADP, turning it into ATP, because we're making a smaller ADP molecule and turning it into a larger ATP molecule. Most of you kind of think of anabolism as building muscles. You're taking amino acids and putting energy in and putting it in to make large muscles. Okay? The opposite, or catabolism, is taking larger molecules, breaking it down to smaller, and that usually yields energy. Okay, somewhat to our exergonic reactions. Okay, breaking down larger glucose molecules into water and carbon dioxide allows us to release energy. It is a catabolic reaction. Okay. So those are a combination of those two determine our metabolism. So that's kind of what we're talking about, breaking down molecules to make energy, or putting molecules together to store or make larger molecules. Usually catabolism drives anabolism. In the case of us breaking down sugar, we're breaking down sugar to drive the anabolic reaction of making ATP. And as we've already talked about, it involves this catabolism, and anabolism involves oxidation reduction reactions. Okay. We're going to start talking about our breakdown of glucose, and we'll see the complete breakdown of glucose is going to require our friend oxygen. Okay. When we are using oxygen, we are doing what we call aerobic cellular respiration. Okay, aerobic with oxygen. Okay, we can also break down glucose doing anaerobic, but we will see this is shortchanged. This doesn't allow us to fully, in anaerobic, we don't fully break down glucose. Okay, so we don't get the full potential of glucose if we're doing anaerobic. Okay, to get the full potential of breaking down glucose, we have to have oxygen to do aerobic cellular respiration. Okay, so this is what we're going to move into. We're going to move into looking at these reactions, but let's first look over this whole process of aerobic cellular respiration. It's actually going to be threefold, 3 steps involved in completely breaking down our glucose. We're going to look at glucose first as an energy source for breaking down, and then we'll look at the fats and proteins, and also a little bit of ketones also. Okay, but let's first look at breakdown of glucose. Okay, so the full aerobic breakdown of glucose, cellular respiration, aerobic cellular respiration, is going to be a 3-step process. Okay, our first step is going to be glycolysis, second will be citric acid cycle, and then we move on to what's called the electron transport chain. Okay, but just an overview of this, and then we're going to break down these steps individually and look at these processes individually, and then put it into the whole process here. But you eat glucose, goes into the blood, okay, and then glucose gets into the cell. Okay, the first process, glycolysis, is the beginning process of breakdown of glucose. Okay. This occurs in cell cytoplasm. Okay, the enzymes for breaking down glucose are in the cytoplasm, and we break down glucose and ultimately end up with pyruvic acid. Okay. This glycolysis can occur without oxygen, so this would be considered anaerobic. We don't need oxygen to allow glycolysis to take place. Okay, and we're going to look at the steps. It's basically multi-step enzymatic process to go from glucose ultimately to 2 molecules of pyruvic acid. Once we're here, if oxygen is available, remember oxygen, aerobic. So if we're doing aerobic, it means that oxygen is available. We can enter into this pyruvic acid that we created, can enter into the citric acid cycle here. Okay, citric acid cycle must have oxygen to take place. Okay, this is considered aerobic. Okay, these next 2 steps, we must have oxygen available for them to run. Glycolysis, we don't have to have oxygen available. Okay, citric acid cycle, where do you see it occurring? After we get to pyruvic acid, we go into the mitochondria. Okay, and you'll see the mitochondria like this bean shaped molecule, and it actually has a little inner, so it actually has 1 and 2 membrane. So where this occurs, citric acid cycle occurs in this inner part of this, inside the second membrane, called the matrix of the mitochondria. Okay, Keanu Reeves style matrix. Okay, electron transport chain, the last process in this aerobic cellular respiration, because again, we need oxygen for this to take place, this electron transport chain. And that is actually going to occur on this inner membrane here. Okay, the electron transport chain. So the first process, glycolysis, takes place in the cytoplasm. The next two, the citric acid cycle and electron transport, take place in the mitochondria. These two must have oxygen. Aerobic glycolysis, no O2 needed. Okay, but you can see if oxygen is not available, we're stuck in just doing glycolysis. Okay, and we're going to see this is not, from an ATP production standpoint, this is not very lucrative. Okay, to get the full benefits of breaking down glucose, we want to have oxygen available. So let's look at this first step in breaking down glucose, glycolysis, and lactic acid pathway. Again, this can occur without oxygen. Okay, we don't have to have O2, oxygen, to run glycolysis and ultimately the lactic acid pathway. So these are considered anaerobic. Okay. Anaerobic without oxygen. So some points of glycolysis. Again, it's first step in breaking down glucose, and we saw this occurs, so the enzymes for these reactions are in the cytoplasm, because this takes place in the cytoplasm of the cell, not in the mitochondria. And ultimately, we're taking glucose as a 6-carbon sugar, and at the end of this pathway, okay, it's not one step to pyruvic acid, but at the end we're going to get 2 molecules of a 3-carbon pyruvic acid. So essentially we're breaking glucose in half. We're breaking it from a 6-carbon into a 3-carbon. So we're breaking it down. We're starting to break down the bonds in the 6-carbon sugar in glucose. Okay, but this is multi-step. There is not just, we go from here to here, we're going to have all these little enzymatic steps. Remember those metabolic pathways in the enzyme lecture. Okay, and at the end of the day, we're going to get loss of 4 hydrogen ions, okay, and we're going to reduce 2 molecules of NAD. Okay, so at the end of the day, we reduce 2 molecules of NAD. What does this mean? Okay, remember reduction. Reduction means NAD is picking up electrons. Okay, remember these are the harvest pickers. They're going to be picking up electrons along the way. We're also going to produce some ATP, which you will see not a bunch. But remember this. Remember, we are reducing. We are picking up 2 pairs of electrons by reducing 2 molecules of NAD. Okay, there are 2 phases of glycolysis. Okay, this is only half of the reactions. This is basically, okay, called the preparatory or activation stage of glycolysis. You can see here's our sugar molecule. And in this first step, we're actually going the reverse direction. We're taking ATP and using it and making ADP. So we're actually breaking down ATP. Remember, the whole goal of this is to ultimately produce ATP for the cell. But in this preparatory phase or activation stage, we're actually using glucose. Okay, or sorry, actually using ATP. Okay, you can see we're actually going to use 2 molecules of ATP are used up. So right now through glycolysis we're negative 2 ATP because we've used them up. We haven't created them when we use them. Okay. At the end of this, we're gonna, and I don't want you carefully remember structures, but we're gonna end up with 2 molecules of basically glyceraldehyde 3. Okay. So as we look at the next steps, remember we have 2 molecules of this going into the next part of glycolysis. So now we're in the energy or conserving, or what we call the payout stage of glycolysis. Here we're actually going to produce some ATP, but what I really want you guys to focus on as we go through these reactions is what's taking place with our NAD. Okay, you can see in these reactions NAD is getting reduced to NADH. Okay, so when we look at this payout stage here, remember at that first stage we actually produced 2 molecules of glyceraldehyde 3. So it's actually 2 3-carbon molecules are going to be going through this step here. Okay, so this reaction here we end up with 2, 2, 2. Okay, so there are 2 molecules moving through. So we'd actually do this twice, but these are the actual numbers of the 2 molecules coming through. We've doubled everything in these, and this little 2 here. Okay, so when we look at this stage, we can see, all right, we reduced 2 molecules of NAD. So we've picked up 2 pairs of electrons. Going from here to here, from NAD to NADH, means we have picked up 2 pairs of electrons in our NAD molecules. Okay, we've now produced 2 ATP. As we go down, okay, we produce 2 more ATP. Okay, so ultimately in this energy payout phase, we've created ATP, but remember we already used up 2 molecules of ATP in that preparatory, those beginning stages of glycolysis. So we netted ourselves a big 2 ATP through glycolysis. Okay, but again, what I want you guys to focus on is what we're doing here, picking up these electrons. Okay, because 2 ATP, if you just do glycolysis, you make 2 ATP, that's not going to fuel your cells for a long period of time. Okay, it's not super efficient because we're not breaking down glucose fully, so we're leaving some energy on the table. So through glycolysis, the net, we get a total of 2 ATP, but we also get 2 NADs reduced. Okay, so we picked up 2 pairs of electrons through this phase here. Okay, because we're going to basically, again, I want you to think of this as harvesting electrons. We're going to go through the citric acid cycle. We're going to pick up more electrons. We're going to be harvesting these electrons. Because we're going to take these electrons and then we're going to use them as energy to make ATP at the end here. So here's our overall takeaway from glycolysis and process. You can see here's that the first steps, we're actually using up ATP, 2 molecules of ATP to activate the glucose molecule, and then the energy or payout phase comes here, and you can see we get 2 ATP, That's 2 ATP, but the net effect is a total of 2 ATP. And we get 2 molecules of NAD reduced. So we picked up 2 pairs of electrons along the way. Okay, I want you to keep track of these because this is going to become the important part, harvesting these electrons as we do these break down these glucose molecules. And at the end we end with 2 pyruvic acid molecules. This is a 3-carbon molecule. We started with the 6-carbon glucose, so essentially we broke the carbon or the glucose in half to 2 pyruvic acid molecules. Okay, so this all occurs without O2, without oxygen. Okay, so this can be considered anaerobic. So when oxygen is available, you saw the full breakdown, those 3 steps occur. We got glycolysis, citric acid cycle, and electron transport chain. When oxygen is available, we can do all 3 of these. Okay, but when oxygen is not available, we are just limited to glycolysis because glycolysis is anaerobic. So when you're working out heavily, at a certain point you're not able, you continue to work out heavily, increase. At a certain point you may not be able to provide oxygen to the muscles. So there is a pathway to get some more ATP without having oxygen available, and that is through basically the lactic acid pathway. Okay. When there's no more oxygen available. Okay. The key is in this molecule here. If you remember, those glucose through glycolysis, okay, 2 of those steps we convert NAD to NADH. All right, the NADH gets reduced. Okay, NADH cannot be re-reduced. It can't keep picking up electrons. It has to give off the electrons, right? It has to be oxidized back to NAD before it can pick up electrons. Okay, and so this has to be available for glycolysis to take place. If we're continually doing glycolysis, there's no oxygen, that NADH is gonna, all our NAD is gonna be converted into the reduced form. We won't have this available. So we can, if we don't have this available, glycolysis stops. So the way to recuperate this is through the lactic acid pathway. Okay, you can see our pyruvic acid, this is what we end up with at glycolysis. And the lactic acid pathway, we go the opposite direction. We are oxidizing NAD. And we reduce pyruvic acid to lactic acid. Okay, but this regenerates our NAD molecules. Now, with this, we can go back, take this to glycolysis, and we can run glycolysis one more time. Okay, so it gives us a way to run glycolysis another time, and that'll yield us 2 more ATP. Okay, through glycolysis, not through this pathway. Okay, through glycolysis. Okay, what this pathway is doing is oxidizing the NADH, so we have NAD available for glycolysis. Okay, but again, it only gets us 2 ATP. This is not going to last you for a long period. If you're working out heavily, it may give you a few more twitches of the muscle, but it's not going to last you for a long period of time. So once you run out of oxygen, you're running out of energy, okay, because you're doing aerobic respiration, and we're stuck doing glycolysis with the yield of 2 ATP. Okay, but this lactic acid pathway allows us to run glycolysis another round. And this is just showing you where, right, this pathway here. this is where we need the oxidized NAD. Okay, this must be available for glycolysis to run. Okay, so this is what the lactic acid pathway is doing. It's supplying us with this, because if we continually run, we're stuck in this. The lactic acid pathway regenerates this, so we can run glycolysis for one more round and get us that big whopping 2 ATP. Alright, we've seen what happens after glycolysis if oxygen's not available. So now let's look at what will happen if oxygen is around and available. We'll finish up our aerobic respiration. Remember, to break down glucose fully, we're going to have the citric acid cycle along with the electron transport chain. Okay. All right, so the full equation of breakdown of glucose is, you can see here, we got our glucose plus O2, and ultimately it'll be broken down to CO2 and H2O. Along the way, we're releasing some energy. Okay. Along the way, we're going to be picking up electrons. Remember our NAD. Our NAD is here, and we'll have one player with the citric acid, another player with the citric acid cycle, the FAD. But these get reduced during these reactions during the citric acid cycle, and we pick up more of these electrons, okay, as these molecules get reduced. Okay, this reaction is similar to combustion, but remember, combustion does the release of energy in one big step, releasing large amounts of heat. Where this, we're doing small enzymatic steps, step by step, just reducing a little bit of, or releasing a little bit of energy as we do these steps. Okay, so we already saw glycolysis. Okay. And at the end of glycolysis, we had 2 molecules of pyruvic acid. We reduced 2 molecules of NADH and 2 ATP. Okay. With oxygen available, the pyruvic acid. Okay. Remember, we have 2 from each molecule of glucose. We'll move into the citric acid cycle. Okay. And remember, we're picking up electrons. We're going to use this electrons later to make ATP. So keep account of the reduced NAD and reduced fat, because those are the important steps when we go to get the big payout for ATP. So there's one step in between, an intermediate step between glycolysis and citric acid cycle, because remember, glycolysis, we did this in the cytoplasm. So now we have that pyruvic acid. Again, each glucose molecule, we get 2 molecules of pyruvic acid. So remember this going forward when we start doing accounting, is that for each glucose molecule, we're going to do 2 rounds of the citric acid cycle. Okay, so you can see pyruvic acid. Pyruvic acid, its fate is it's going to be, as it moves in to the mitochondrial makeup, it gets shipped in. Okay, a molecule of CO2 is being produced. Okay. You can see pyruvic acid is 3 carbons. When ultimately we're going to convert pyruvic acid to acetyl-CoA. First, we're going to remove a carbon. Whenever we remove a carbon during the citric acid cycle, we produce CO2. Okay. This step also reduces one more molecule of NAD per pyruvic acid. For glucose, then, we reduce 2 molecules, because remember, 2 molecules of pyruvic acid are created from each glucose in glycolysis. Okay, so we went from a 3-carbon to 2-carbon, and then we added coenzyme A in this step. And the end of this is acetyl-CoA. Okay, remember this molecule, because this molecule is central to the metabolism. It's kind of the bridge between fatty acid metabolism, protein metabolism, and glucose metabolism. Okay, so acetyl-CoA is kind of the unifying molecule between those metabolisms of these other nutrients. Okay, but now we've gone pyruvic acid converted to acetyl-CoA. We produced 2 NADH, or reduced 2 molecules here. Okay, if you remember glycolysis, we already reduced 2 molecules of NADH. Okay, we have not produced, and through glycolysis we also did 2 ATP. So for keeping track of what's going on, here's what we got so far: energy, or at least what's going to lead to energy. Okay, glycolysis, we got 2 ATP. We had 2 ATP that we create, or 2 NADH got reduced. We picked up 2 electrons during glycolysis, and now through this step, this is kind of the step people miss when they're counting their NADH, the reduced NAD. But in this step, going from pyruvic acid to acetyl-CoA, we've created 2 molecules, or reduced 2 molecules of NAD to NADH. Here's just looking at that step closer. There's that pyruvic acid. Remember, we got 2 of these from glucose, and here is coenzyme A. Okay, so we're gonna tear off 1 carbon. So we're going from a 3-carbon to a 2-carbon, because we produce that CO2, and then we add coenzyme A to that 2-carbon molecule. Okay, and you see that there. And do this reaction. All right, we reduce NAD to NADH. So now we pick 2 more reduced NADs. We picked up 2 more pairs of electrons. All right, so I said acetyl-CoA is the link between glycolysis and citric acid cycle, because acetyl-CoA is what actually enters the citric acid cycle. Remember, this is a 2-carbon molecule. Okay. And to start the citric acid cycle, you will see, and it's also called the TCA, tricarboxyl acid cycle, or the Krebs cycle. I will use citric acid cycle, the name. I believe that's what your book is using. Okay, but to start out, you can see this is going to be basically a cycle. We're going to start here with oxaloacetic acid. So I'm not going to have you memorize all these intermediate molecules through the step, but I kind of have you guys remember these beginning molecules, and ultimately I want you to know what we are getting out of the citric acid cycle. Okay, but the start is, we got oxaloacetic acid is a 4-carbon molecule. Acetyl-CoA, 2-carbon. These 2 combine to start the citric acid cycle. When they combine, they form citric acid. Okay, we'll look at this a little bit closer, but through one cycle of a citric acid cycle, you can see as you pull off carbons, this is where we're going to be producing our CO2. So our CO2 gets produced through this converting pyruvic acid to acetyl-CoA, and then also in the citric acid cycle. You saw we produced no carbon dioxide in glycolysis. So if you're doing anaerobic reaction, you're not producing CO2, but if you're going full aerobic reaction, you're going to start producing the CO2. Okay. Here is what is coming out for each acetyl-CoA entering the citric acid cycle. 3 molecules of NADH are being reduced. 1 molecule now of FAD is being reduced, and we're forming 1 ATP. So if you think of this in the terms of glucose, you would have to double these numbers. Okay, because how many acetyl-CoAs are formed from 1 glucose molecule? We have 2. Okay, so you can double these numbers. This is just one cycle through. 1 acetyl-CoA, 1 cycle through the citric acid cycle. Okay, but for glucose, when we start doing the lipids, we're talking 1 cycle through with acetyl-CoA. Okay, but for glucose, since we formed 2 acetyl-CoA, we would double these numbers for each glucose molecule. So important events, I'm not going to really be worried about you knowing GDP, but ultimately there is, for each acetyl-CoA entering, you form a molecule of ATP. You reduce 3 NAD and you reduce 1 FAD. Okay, that is what is coming out. Also, you saw that we produce CO2 along the way. All right, so looking a little bit closer at the citric acid cycle, you can see here we are, we're bringing in acetyl-CoA, and we start with oxaloacetic acid, that 4-carbon molecule, and they combine to make citric acid cycle to start the process. All right, so you can see all these intermediate steps along the way, but what you need to know from these steps is, okay, for each glucose, we formed 2 NADH, or reduced 2 molecules of 6 molecules of NAD, reduced 2 molecules of FAD, produced 2 ATP, and also produced 4 CO2 molecules. Okay, if we were doing our calculations of our ATP and also keeping track of our NAD formed or reduced, we now have 4 NAD reduced by the time we got to citric acid cycle through glycolysis and converting pyruvic acid to acetyl-CoA. Okay, and we also formed 2 ATP from glycolysis net, because remember we put in 2, made 4, but we had a net of 2 ATP from glycolysis. So if we put these together, we end up with 6 plus 4, we end up with 10 NAD molecules reduced, 2 FAD molecules reduced, and 4 ATP formed. All right, so we've done all these reactions and we formed a measly 4 ATP. That isn't going to keep our muscles or our highly active cells going for long periods of time. So the key is that we did this. We reduced NAD and we reduced FAD. Okay, and when we reduced these, what do? We picked up pairs of electrons. Okay, because we're going to move those electrons forward. These molecules are going to get oxidized and give their electrons off to another molecule. Okay, and we're going to use that electron to do some form of work. Okay, when we get to the electron transport chain. So this is the key, is that we did harvest these electrons, because we're going to use energy from these electrons to do some work to give us a bigger payout of ATP. Alright, so here's where we're going to get our big payout of ATP, and here's the reason why we basically were reducing those molecules of NAD and FAD is to use here at the electron transport chain. Okay, I'll draw our little, you remember we're using our mitochondria, and our mitochondria has basically these inner membrane with all these folds. Okay, here is where the electron transport chain is going to take place, on the membrane of this inner membrane of the mitochondria. So remember, the citric acid cycle takes place in the matrix. It takes place in here. So all those NADH, all those FAD are sitting in here with their electrons that they picked up. Okay, and we're going to look at, there are several proteins involved in this electron transport chain, and they are located on this inner membrane. Okay, so what's going to happen? Basically, electrons are going to get transported. Okay, electron transports molecules, they pass electrons down the chain. And we're going to see molecules are being reduced and then oxidized. Reduced, so it is a series of reduction and oxidation reactions that move the electrons through the transport chain. Okay. As we move through, they are exergonic reactions, and we're going to use these electrons ultimately to make ATP. We'll see we're going to use these electrons to do what we call pumping protons. Okay, and ultimately that is going to be used to make our ATP. Okay, ADP is phosphorylated. This process formed down the electron transport chain, taking electrons, oxidation, and causing phosphorylation. So it's called oxidative phosphorylation here. It's not 100% because in our reactions usually heat is released. But we will get a much greater payout of ATP than we have from the previous two steps. So I'm not going to have you memorize names here. I want you to realize what is here at the electron transport chain, because I don't want you to get lost in the names. I want you to understand the process that is taking place here. But you will find there are 4 protein complexes. Okay, you can see one here, one here, one here, one here. I'll call them complex 1, 2, 3, and 4. Okay. At the end of this, there is this large enzyme, called ATP synthase. This is where our ATP is going to be produced. Remember, we take ADP, add a phosphate group to that second phosphate group of ADP, and create ATP. Okay, but this is where the electrons are going to be passed. Okay, you can see our NAD, our NADH. This has electrons, and it's going to get oxidized at complex 1. Now complex 1 is reduced. It has picked up the electrons. Okay, and complex 1 is going to transfer electrons to complex 2, and complex 2 is like a little shuttle between 1 and 2, and it's going to, so now complex 2 is reduced, complex 1 is now oxidized. Now complex 2 is going to go pass its electrons to complex 3. Complex 3 is now reduced and complex 2 gets oxidized. There's another shuttle between complex 3 and complex 4, but electrons are passed off, and ultimately complex 4 gets reduced and complex 3 is oxidized. And then we will get to ATP synthase. But let's look at what is taking place as we pass these electrons through. Okay, there is work being done as these electrons are being passed from these complexes here. This here just shows what's taking place for the transport of electrons. Remember, it is a series of reduction-oxidation reactions that are passing the electrons through here. Okay. One other thing to remember is when one of these complexes is already reduced, it can't now accept more electrons. It has to be oxidized before the electrons can accept more electrons. Okay. And the electrons pass through in this direction, not back and forth from 1 to 2 to 3 to 4. Okay. So what is those electrons? What is the energy being used to do? Look at this complex, not complex 2, but complex 3 and complex 4. Okay. The passing of the electrons, the energy from the passing of the electrons is being used to pump protons. Okay, remember our little mitochondria. Okay, our electron transport chain is taking place in this inner membrane. And so what is taking place as we pump electrons is, or pump protons, sorry, is the protons are getting pumped into this inner membrane space between the two membranes. So what's going to happen, what's occurring is we're going to have this large concentration of hydrogen ions. We're creating a hydrogen ion gradient. Because if you remember, we saw with secondary active transport, these gradients have energy. They contain energy, potential energy. Okay, because we're going to use that potential energy finally to make ATP. Okay, so if you want to do like an overall picture of this, you can think during glycolysis, during citric acid cycle, we were harvesting these electrons. We were reducing NAD and reducing FAD. We were picking up electrons along the way. Then we passed the electrons to the electron transport chain. The passing of electrons is causing protons to be pumped into the inner membrane space, making a concentration gradient of protons. Protons are high here and will be low inside the matrix. Okay, so now we have ourselves a proton gradient between this inner membrane space and the space inside the matrix. So here's where the process, you can see why we are creating this proton gradient. Because if you remember at the end, we had that big ATP synthase, and that's what you see here. Okay. This is what we call the chemoosmotic theory. This is coupling the protein gradient formed with the creation of ATP. And what's happening is this proton gradient, lots of protons in the inner membrane space, very little in the matrix. As these protons, if they're allowed to, they're going to want to move from the space into here. And how they are going to do that is they are going to move through this ATP synthase. And for each 3 protons that move through, that's going to be enough energy to catalyze the reaction of ADP to ATP. Okay, so that protein gradient is being used, that energy there is being used to fuel that inorganic reaction, that ADP to ATP. Okay, so I'm going to post a video, a link for a video for you to watch this. It's pretty cool on watching how this protons move through and we get catalyzation of ATP. Okay, but without a proton gradient, this will not get made. Okay, and so to keep this whole thing running, this electron transport chain, because remember, this is aerobic respiration. We needed oxygen to allow this to take place. So why is that? Okay, we've done our citric acid cycle, we needed oxygen, and our electron transport chain, we're going to need oxygen for this to continually take place. So let's see what the function of oxygen is in this case. All right, so let's look at the function of oxygen here. Oxygen's main function, or its role, is to be a final electron acceptor at the electron transport chain. Okay, remember these electrons are getting passed along as we go through. And ultimately, when the electrons get to complex 4, the last complex in the chain, oxygen picks up the electrons from complex 4 and is converted to water. Okay, in that reaction there. Why is this important? Because remember, those electrons are getting passed through here from oxidation reduction reactions. When complex 4 is reduced, it cannot accept more electrons, all right, until it is oxidized. So who is doing the oxidation of complex 4? It is water is picking up the, or sorry, oxygen is picking up the electrons and being formed of water. So now this complex is oxidized, it can pick up electrons. If oxygen is not here, complex 4 stays reduced. The little shuttle stays reduced. Complex 3 stays reduced, complex 2 stays reduced, and complex 1 stays reduced. If these complexes are not oxidized, then NADH cannot drop off its electrons. Okay? When this complex is reduced, it cannot pick up more electrons. Okay, so without oxygen, this electron transport chain gets backed up. We're not transporting electrons through here. We're not pumping the protons. Okay, if we're not pumping the protons, we're not making this proton gradient. And if we don't have the proton gradient, we're not able to use ATP synthase. Okay, so this is why oxygen is important for aerobic respiration, for us to go through citric acid cycle and through the electron transport chain, because it picks up these electrons to keep the flow of electrons going through the electron transport chain. Okay? Without it, it's all backed up. We're not creating that proton gradient, and we're not able to use ATP synthase, and there it goes. Okay. Our balance sheet after we're done with the electron transport chain through our oxidative phosphorylation. Okay. During our glycolysis and our citric acid cycle, it's called at the substrate level, we have created 4 ATP. Okay. Through the substrate level, or direct phosphorylation, not through oxidative phosphorylation. Oxidative phosphorylation takes place at the electron transport chain. Okay, in the oxidative phosphorylation, theoretically, and we're going to look at these theoretical yields, we get 3 ATP for each NADH molecule and 2 ATP for every FAD molecule that drops its electrons. When these take their electrons to the electron transport chain, what is the yields? For each NAD, you can yield 3 ATP at the ATP synthase, and for each FAD electrons brought into the electron transport chain, it can relate to creating 2 ATP molecules. Well, theoretically, we can yield basically 36 to 38 glucose molecules, because we'll pick up 32 to 34 at the electron transport chain. So remember, we had 10 of these coming in, and we have 2 of these. Okay, so 3 times 10, 2 times 2 equals 34. That would ultimately, with these 4 here, would give us 38 ATP. Okay, so you can see how aerobic respiration is allowing us to create a lot more ATP. Allows us to do work. If you're doing workout and your muscles are working, you're going to do a whole lot better if you have 38 ATP rather than 4. That happens when you do a round of anaerobic respiration. Okay, so oxygen is important to be able to constantly produce a significant number of ATP. Okay, here's the numbers we'll be using. I'm going to have you guys use actual yields for what is created by NAD and the reduced FAD. So for actual yields, because this takes in energy needed to move ATP out of the mitochondria and moving other molecules in, okay, is for every pair of electrons brought to the electron transport chain by NAD, you can yield 2.5 ATP. For every FAD molecule electrons brought to the electron transport chain by FAD, you can yield 1.5 ATP. So again, remember, for glucose, we ended up reducing 10 NAD and we reduced 2 FAD. Oops, FAD. Okay. So times this by 2.5 ATP, times this by 1.5, and what's that end you up with? 10 times 2.5, 25, plus 2 times 1.5 is 3, so we'd end up with 28 ATP. And if you remember, this would be 28 ATP by oxidative phosphorylation at the electron transport chain. And we also, during glycolysis and citric acid cycle, we made 4 ATP. So that would ultimately, actual yield would get us around 32 ATP, what we see up here. Okay. So this is what I'll be using. So that theoretical yield was 3 and 2. We will be using the 2.5 1.5 when we're trying to calculate how much ATP is formed through oxidative phosphorylation from these electrons from NAD or FAD. Here's the accounting done here. This is better since it doesn't have my scribble with my PowerPoint little scribbling. You need to give me a little text part here, but this is the numbers we're looking at. See, this is the total outcome that we see there. The actual yield from the NAD or FAD when we bring it into the electron transport chain. Okay. So we've seen how glucose can be converted into ATP, but let's kind of relate this now in an overall bigger picture of metabolism. Okay, because we can store glucose when we need it. If for some reason we have to fast, we have a store of glucose that we can convert back or convert into glucose that is usable to make ATP. And that storage of glucose is going to be glycogen. Okay, your liver and your skeletal muscles can store glucose in this large chain. Basically, it'll be, there's a glucose molecule, glucose, and it will branch off, glucose, but this large storage chain is called glycogen. So for osmotic reasons, the cell can't just store glucose as a single molecule. So when to store glucose, it makes what I showed you, that long chain of glucose. And making that long chain is called glycogenesis. Okay, when sugar is, or when nutrients are high, all right, we don't have to use that glucose for energy right away. We can go ahead and store it away in glycogenesis. Although, if you think about it, there is not a huge store of this glycogen. Okay, we can't just constantly keep building glycogen. And that's kind of where that comes into play. We start, after we've replenished our glycogen stores in our liver and our skeletal muscles and also our cardiac muscles, it would be shunted to fat. But say if you had been fasting for a long period of time, you probably have used up some stores of glycogen. Okay, you start eating again, you can replenish the glycogen and have a nice fast ability to get to glucose by breaking down glycogen. Glycogen's formed from glucose. It's called glycogenesis. Okay, we'll see glucose is phosphorylated, and then an enzyme called glycogen synthase starts joining the glucoses together. Okay, and so then when the glucose is needed, we can do glycogenolysis, breaking down of glucose. You can kind of see the flow of what's taking place. When there is lots of glucose, okay, we have an oversupply, all right, but we still haven't created all our glycogen. We can move glucose in. Okay, this, I need to make the caveat, is the liver only. Okay, this is the liver. Okay, we have an excess glucose, we can bring it in and it gets phosphorylated, but then ultimately gets formed into glycogen. Now we've been fasting, our blood glucose is going low. We get glucagon comes in and tells our liver, hey, we need some glucose in the blood. Now we can go in this direction and we get the glucose 1-phosphate and glucose 6, and the liver pulls off that phosphate and can release glucose into the blood. If the cell needs energy, then the glycogen can be moved and brought into glycolysis in the cell. Okay. Remember, the muscle tissues, the cardiac and skeletal muscles, also convert glucose to glycogen. They have a store of glycogen. Okay, but the difference between the liver and the muscles is the liver is the only tissue that can actually release glucose in the blood. The glycogen that is stored in the muscles ultimately goes down in this direction. Okay, the glycogen stored in the muscles is used by the muscles. The muscles will not release glucose into the blood. The only organ that does is the liver, okay, because the liver can remove that phosphate that is still here on glucose 6-phosphate to release the glucose into the bloodstream. Okay, so again, overview: liver, skeletal muscle, cardiac muscles have stores of glycogen, have these stores of glucose, but the muscles will use, if it needed, will use it for its own energy production, where the liver can produce or release glucose into the bloodstream. Alright, so we've kind of gone through our sugar and the metabolism of the glucose or the carbohydrates. So we do have other molecules we can use for energy sources. Lipids, so our fats, our fatty acids, and proteins. Typically proteins will be the last to go after because we have proteins in the muscles. We want our muscles. We don't want to be breaking them down, but when sugar is not available, we can start breaking down lipids, and at last resort, we can break down proteins if needed. And if you remember, there was one molecule that is going to be the unifying molecule between glucose, lipids, proteins, and that's going to be that acetyl-CoA. Okay, remember acetyl-CoA? Where do we find it? It is at the beginning of the citric acid cycle. Okay. So as I said, we can use lipids and proteins as energy if needed, but many times in our society, when there's more food. Okay, I told you ATP we're not constantly making ATP because we can't store it. ATP will just, if it's stored for long periods, will go back from ATP to ADP. So now we used all this energy to make ATP and we store it and it just reverts back. So we don't keep these long stores of ATP, so we store our energy. We already saw we can store it as glycogen. Once we have glycogen stored away, we can start reverting that store into using fat as storage. Okay, so we don't use ATP. We can shut off and we can store energy as glycogen, or after glycogen we start storing it away as fat because we can now use that fat later or those lipids to create ATP when needed. Alright, so we have an excess of, say, glucose. So what happens as we go down? You can see here is our glycolysis pathway. Okay, we got our glucose coming in, and if we have an excess amount of glucose, we're going to shut down. We don't need to do the citric acid cycle. We don't need to go here because we don't need to make ATP. We got enough ATP made, but we still have excess glucose. We can, instead of going down glycolysis, we can revert and create glycogen. Okay, if glycogen is stored away, we can come down the glycolysis pathway, but instead of reaching into the citric acid cycle, the acetyl-CoA is going to be shunted into making fatty acids. the 3-phosphoglyceraldehyde is going to make that glycerol backbone. Remember, our lipids are glycerol, and then there are 3 fatty acids attached to that glycerol backbone. So here we can shunt the products of glycolysis into converting them into fat for later storage. Okay, one thing to remember, acetyl-CoA, basically, instead of putting strings of glucose together, we're going to be putting strings of acetyl-CoA to make our fatty acids. Acetyl-CoA was a 2-carbon. So when we look at our our fatty acid chains, they're going to be typically even number because we keep putting acetyl-CoAs together to make a chain of fatty acids. Okay, so you'll see stuff like 14 carbon, 18 carbon, okay, because we've been adding 2 carbons at a time when we're making our fatty acids from acetyl-CoA. Okay, here's just kind of explaining that slide, the previous slide, and what you talked to about shunting the glucose to glycogen, or shunting those products of glycolysis into fatty acids to form the fat. And here's kind of how we look at this when we start looking at acetyl-CoA. Okay, I told you this is the unifying molecule. Okay, if we need energy, we can bring acetyl-CoA to the citric acid cycle. Okay, we can also make steroids, cholesterol, and so forth. One you guys have probably seen is ketone bodies. Okay, in our ketogenic diet, or if we have lots of energy needs have been met with the blue, we can shunt them to fatty acids and store that energy away. Okay, but the key is this is the unifying molecule where we need to go. If we need energy, we can break down fat. We're going to see we can break down fats to acetyl-CoA now, and we can move those into citric acid cycle. We can also break down proteins, form acetyl-CoA to bring in the citric acid cycle. Okay, so we'll see this is in the core, or the unifying factor in our metabolism throughout our body. So just as we stored away glycogen, we can store away fat, and we store it away in these triglycerides in our white adipose tissue, our white fat. Okay, these are good stores because when we need energy, we can do lipolysis. We can break down the triglycerides, and then we can basically, as we break them down, we're going to be converting them back into acetyl-CoA. Okay, so if energy stores are not met, we've been fasting, we need some energy, we need to create the ATP. We can break down these fatty acids and use them as energy to make our ATP, but we'll be breaking down to acetyl-CoA. So here is what we call beta oxidation. Beta oxidation is breaking down the fats to make energy stores. Remember, these fatty acids we created were even. Okay, we created it through acetyl-CoA, putting acetyl-CoA molecules together. Now we're gonna do lipolysis. We're gonna break down these fatty acids through beta oxidation. And you can see, you don't need to know the steps along the way here, but you can see we're going to be, each time we're going to be taking off 2 carbons from those fatty acid chains. Okay, but I want you to look at the reactions here. Okay, we had to put some ATP in. So each time we cleave off 2 carbons, we have to use up a molecule of ATP. But as we go through to cleave off the acetyl-CoA. We are oxidizing the ends of what's left of the fatty acid chain, and we're creating, or we're reducing FAD, and we're reducing NAD through this chain. So each time we create a acetyl-CoA from a fatty acid chain, you can see, remember FAD, if we took FAD, the electrons from FAD to the electron transport chain, we would get 1.5 ATP. If we took NAD's electrons to the electron transport chains, those can be converted to 2.5 ATP. So each time we cleave this, we are making 4 minus 1. So we can get 3 ATP for each time we cleave off 2 carbons from our fatty acid chain. But what else do we create each time we cleave off 2 carbons? We're making a molecule of acetyl-CoA. At the end of the day, acetyl-CoA, you remember we make 3 NADH for each acetyl-CoA, 1 FAD H2, and we create 1 ATP. Okay, this isn't like glucose. Remember, for glucose we brought in 2 acetyl-CoA. We're just saying how much is each acetyl-CoA going into the citric acid cycle worth. And if you remember, this is worth 2.5, this is worth 1.5, and we created 1 ATP. So ultimately, through the citric acid cycle of acetyl-CoA, we yield 10 ATP. Okay, so each time we cleave, boom, we can convert that to 3 ATP. Each time we cleave, each cleavage we make acetyl-CoA, and that is worth 10 ATP. So let's look at a 16 carbon fatty acid. Let's go to the next slide. So let's look at, again, a 16 carbon, breaking down a 16 carbon fatty acid. How many acetyl-CoA can we yield from this? Each acetyl-CoA is 2, so 16 divided by 2, 8 acetyl-CoA. This is not drawing well. Okay, and how much are each of these worth at the electron transport chain eventually? 10 each. 10 ATP from each of these acetyl-CoA is going into the citric acid cycle. So we come up with 80 ATP. Okay. The key is if you remember through these reactions, as we beta oxidize, each time we cleave off, we yielded 3 ATP. The key is at the last carbon, there's nothing left to oxidize. So we actually do 1 less cleavage than we make of acetyl-CoA. So we only do 7. So 3 times 7 equals 21. Okay, so you guys have this in your slide. I want you to scratch this out, because what I didn't take into account is that through those reactions you had to put in 1 ATP. So instead of getting 4 out, you essentially get 3 out of each cleavage. 3 ATP are produced per each cleavage. Okay, so if you want to figure this out, like an 18 carbon, you would have 9-acetyl-CoA, and you would have 8 cleavages. All right, you would cleave off to 8 or oxidize the ends 8 times when you cleave, because you do 1 less than the acetyl-CoA produced, and so your calculation is 9 times 10, 8 times 3. And that'll get you how much ATP. So you can see this lipids is a good storage. You can produce lots of ATP from each of these fatty acid chains. So you can see we can get almost 3 times as much ATP from one molecule, one fatty acid, than from one molecule of glucose. And therefore you have to exercise quite extensively to use up this ATP to break down this fat. So you see how it's harder to break down fat than it is glucose there, or at least energy wise. One other molecule we can use, all right, to produce ATP, we got ketone bodies. Your body can use ketones. All right, this is the premise of the ketogenic diet. If people that are on the ketogenic diet are basically using ketones for energy instead of sugar. They're eating very, very low carbohydrates. And so what happens in this case, you eat high fat, your body starts increasing fatty acid, starts breaking down fats. If there's a lot of fatty acids in the blood, the liver will go ahead and convert those fatty acids into ketone bodies. Okay, you see acetone, that's kind of the smell you get. You get an acetone smell when you start doing ketogenic diet in your breath. Acetoacetic acid and beta hydroxybutyric acid. So you can see these are acidic. So if you start doing ketogenic diet and start producing lots of ketones, what do you think would happen to your blood pH? Yeah, they're acids, so your blood pH would actually go down. Not to a pathological level, but your pH would be typically lower than if you were just eating a regular old diet. Okay, what happens is these ketone bodies, what do you think they get converted to make energy? Yeah, that unifying molecule will be converted to acetyl-CoA, and then the acetyl-CoA can enter the citric acid cycle, and we can produce our ATP. Okay. We won't do any accounting here number-wise, like our lipids, but just realize ketone bodies get converted to acetyl-CoA, and then that can enter the citric acid cycle. Because we'll look at protein breakdown in a second, and where do you think the molecule, ultimately, if we need proteins for energy, they're going to be converted to, again, acetyl-CoA. That is the unifying molecule for the metabolism. So if need be, amino acids can be used for energy, or if we have an excess amount of amino acids, they can also be converted to glycogen and to fats. Okay. So we'll see our bodies. We can make 12 of the 20 amino acids that are out there. Okay. So there are 8 that we cannot make. Those would be, you probably have heard, non-essential and essential amino acids. The 12 that we can make are non-essential since we can basically create them in the body. But there are 8 that we have to basically get in our diet. We cannot make them. So they have to, if we need an essential amino acid, we have to make sure we have it there from the diet. Okay, we can't be making them. You don't have to memorize all the amino acids, okay, but you can see here are the 12 that we can make. Here are the 8. In children, actually, there are 9. They have to get histidine. We can actually make this in adults. Okay, but that is the difference between essential and non-essential, is these are ones the body can make from other molecules, where these essential amino acids we have to get from our diet. So how do we make these amino acids? These non-essential ones is, you see, a process called transamination. And many of the amino acids are going to be made from basically those intermediates in the citric acid cycle. Okay, these intermediates, the pyruvic acid and the intermediates, remember we had all those molecules throughout the citric acid cycle that I didn't make you memorize, but they are called keto acids. And if we can add an amine group to some of these intermediates in the citric acid cycle, we can basically create those non-essential amino acids. Okay. We look at this transamination process. Okay, and each one requires a specific enzyme. But basically what we're going to do is remove an amino group from one amino acid and move it over to one of these keto acids to create one of those non-essential amino acids that we can make in the body. Okay. Looking at the transamination, you see glutamic acid. Basically, what are we doing here? This should look familiar. Oxaloacetic acid, that is the molecule that gets bound with acetyl-CoA to start the citric acid cycle. But you can see here, all we're going to do is swap the amine group from the glutamic acid and bring it on to oxaloacetic acid. When we add the amine group to the oxaloacetic acid, that actually produces one of our non-essential amino acids, aspartic acid. Okay. Same, you can see kind of the molecule that is going to be the donator or carrier of the amine groups is glutamic acid. Okay. Again, we're swapping the amine to a molecule of pyruvic acid, one you should be familiar with, right? And when that gets added, we actually create alanine, another non-essential amino acid. So these transamination processes allow us to create those non-essential amino acids. So now you can see how amino acids kind of lie in our energy production. If we needed to, we could use these. You can see the different amino acids. We can bring them into a citric acid cycle. Ultimately, we get acetyl-CoA converted and we can bring it in. If we have a glutton of these, if we have too much of these, you can see we can get converted to pyruvic acid, acetyl-CoA, and we can shunt this just like we did with excess glucose into making fat production. Okay, so depending on what the energy needs are, if we low on energy and we're breaking down fats, we're breaking down proteins, and we have these amino acids, we can use them for energy. But many times, if we have an excess of them and we have ATP already produced, we can shunt these up to making fat stored for later. Okay, so you can see how this is all kind of intertwined, all these nutrients, because ultimately they can all get converted to acetyl-CoA, and then depending on energy needs, we can go either use it for energy or use it acetyl-CoA to store in fats for later for use. So that's it for this. Then we're going to be moving on to the nervous system. So make sure you got your membrane transport down, because we will be using lots of membrane transport for our nervous system.